Welcome to Past Medical History, a series of conversations brought to you by the BJCA in partnership with Wondr Medical. We hope to inspire you with these stories that have influenced modern cardiology practice, brought to you directly from the people who made those changes. I'm honoured today to be joined by Sir Rory Collins, who's the BHF Professor of Medicine and Epidemiology at the University of Oxford. Sir Rory's impact on cardiology has been profound, with his work demonstrating the value of thrombolysis in acute MI and the value of statin therapy affecting the way that we practice medicine globally today. His work has been recognised with Lifetime Achievement Awards from the British Cardiovascular Society, the European Society of Cardiology and the Medical Research Council. He currently serves as the Principal Investigator for the Biobank UK and as the head of the Nuffield Department of Population Health. So Sir Rory, it's really great to have you here today to share your stories with us. Thank you.
I'm looking forward to it. Thank you very much for inviting me.
Great. So megatrials today are now an established research tool that we use in cardiology, with studies of thousands of patients presented as late breakers at all the big conferences. And I think you can be credited with some of the earliest, namely ISIS-2. I want to take you back to before those studies, and I was wondering if you could explain how research informed clinical practice and how the studies that were done before that were implemented clinically.
Well, I came into clinical trials in a rather roundabout way. I had wanted to do natural sciences at Cambridge. I thought I'd like to do mathematics and then go into medicine, but didn't manage to get in. So I went to St Thomas's and did my preclinical there. And in the second year, I thought, well, I wonder whether I could do some more mathematics. As you know, you can do an intercalated BSc. And I said, well, could I do one in statistics? And apparently no one had ever asked to do that. You could do it in psychology, history of medicine, anatomy, physiology, biochemistry, whatever, but not statistics. And I went around London University to see if anybody would take me on, and no one would. So I went to the library and found the addresses of 36 US universities, because I'd heard America was more flexible. And I wrote to them all, and one of them said, yeah, you can come and do a degree in a third year in statistics. So I went to George Washington University in 1976. And the professor of community medicine, Walter Holland, heard about this and said, can I help? And I said, well, I've got no money to go and do this. The MRC and the medical school are not supporting it. And so he said, well, I happen to know the person who set up the cancer clinical trials in the National Cancer Institute, Bernie Cornfield, and he now runs a clinical trials unit, biostatistics unit, as well as being professor of statistics at George Washington. And they offered me a job working 1 day a week, which helped pay my fees. But it was working on a National Institute of Health clinical trial on the dissolution of gallstones with chenodeoxycholic acid. And so I was working on this randomized trial. And I suddenly realized that this was how you were meant to evaluate treatments, that you randomized patients. And so at the end of the year, I thought, I really want to do more of this. So I applied to the London School of Hygiene and Tropical Medicine to do a master's in statistics and got accepted. Came back, and then they realized I was trying to do my clinical training my last 2 years while also doing a degree at the London School of Hygiene and Tropical Medicine. So I was kicked off the London School of Hygiene course, but I was allowed to go to the lectures. I just couldn't do the degree. And then I completed my medical school training, was doing my house jobs, and I thought I'd really like to do some research. And I'd read this book by Peter Armitage on basic statistics, and I just rang him up at the London School. He had moved, he was in Oxford, and so I arranged to go and meet him, and I was talking to him about I wanted to work in clinical trials, and he said, well, I'm an applied statistician, but I don't actually do them, but you should talk to Richard Peto. And he was one of the kind of names I knew of. And so he rang up Richard, and I could hear Richard Peto saying, I'm too busy. But he can come over and make an appointment to meet with me. So I went over onto the old Radcliffe Infirmary Hospital site and found the corridor where they all worked. And there was this man fixing a chair in the corridor. And I was looking around. He said, "Are you looking for somebody?" I said, "I'm looking for Richard Pito's secretary." And I said, so he said, "Well, I'm Richard Pito. What do you want?" And so he took me into his office, and I started talking about what I wanted to do, and he started talking about clinical trials. He then gave me some things to read. And it was really Richard. Richard had this idea that the problem with the trials of the past was that they were randomizing. Austin Bradford Hill had introduced the first randomized trial with the MRC of streptomycin in TB, but it was a small randomized trial. The treatment was fantastically effective, so it demonstrated the benefit of the treatment. But you could have known that without randomizing. And so unfortunately what it did, it developed a standard of randomization, but small trials that just weren't big enough to detect a lot of the treatment effects that existed. And it was Richard that was really arguing that you needed to do randomization in order to avoid bias, but you also needed to have much larger numbers in order to reduce random error. So you got rid of systematic error by randomizing, large numbers to get rid of random error, and the two together were critical. And that's what he was writing about back in the 70s. But he couldn't find anybody to do it. And the way it came about was Peter Sleight had this incredibly smart PhD student, Salim Yusuf, working with him. And they had been doing some early studies, animal studies initially, looking at whether you could reduce infarct size with beta blockade. And they had some promising results, and Peter knew Richard Pito, so he went down and with Salim and said, "We want to do a randomized trial to see whether in humans giving intravenous atenolol would reduce infarct size." And so they talked about designing a trial and how big it needed to be to look at surrogate markers on ECG and things like that. And they did that study and it looked quite promising. And so then they started talking about, well, what would you need to do to show that that had effects on mortality? And this is when I think Richard realized that he had people who wanted to do the right thing. There was a guy called John Cruikshank at ICI who was interested in trying to help make this happen. So between the cardiology department with Peter Sleight, who was head of cardiology, funded by the British Heart Foundation, with Salim, who had finished his PhD, with Richard from the statistical side, and with the backing of what was Stewart Pharmaceuticals, became part of ICI with John Cruikshank, they cooked up this idea of actually being able to do a trial with several thousand people, the ISIS-1 trial, of intravenous beta blockade followed by oral beta blockade, to see whether the animal studies that showed you could reduce infarct size, the small-scale human studies that showed improvement in surrogates such as the mapped size of the infarct, arrhythmias, turned into clinical benefit. And that's when I came in the door. So I went and finished my house jobs in Stoke-on-Trent and came down to Oxford in 1981 to work on this first ISIS-1 trial for a year. I thought I'd do research for a year and learn about how you deliver trials. And with the backing and the support of Stewart Pharmaceuticals, they introduced us to people in Scandinavia, then in Australia, and this trial spread out from the UK into these other countries and into Italy, and kind of sold the idea of a collaboration between clinicians, not academic, but clinicians in the local hospitals who were thinking, well, what do I do with my heart attack patients? And randomizing them. They would ring up our coronary care unit here in Oxford, and they would give a few details, the name, a little bit about the ECG and the symptoms and the time of onset, and then they would get told whether to give the beta blocker or open control. And that's how it all started. And after the first year, I thought, well, do I really want to leave? And I thought, well, I'll do that master's now. So I did the master's in statistics with Peter Armitage here in Oxford. I thought at least, well, after 2 years, I'll be able to go back to clinical medicine. At least I can say I've got a degree out of it. And then we started thinking, well, so what next? And one of the things that Richard had really pioneered, along with Tom Chalmers in Boston, was the idea that, okay, you have a large number of small trials, but could you combine them? If you've got 20 trials of a particular treatment, each of which on its own is too small, could you combine them and actually get a clear idea about what the treatment does? And so Salim and I started to look at what trials have been done for the emergency treatment of MI, with what were all the treatments that had been done. And we just collected all the data from the published papers for all the different treatments. And the one for which there was the most evidence was intravenous fibrinolytic therapy, largely streptokinase, but also some urokinase. And they were kind of 24 or 48-hour treatment regimes. We got the publications and we wrote to all of the people who'd run those studies. We asked them for some more detail, more data. They were incredibly helpful. I remember going to a meeting. I was in Australia for ISIS-1 and going to the hospital where they'd run one of these trials, and he just let me look through all his records and all his data and work out what the numbers were. And Salim and I used to go to his spare bedroom at a weekend, and we would work through this data, and we would then we kind of started honing down on intravenous fibrinolytic therapy, and it just looked really promising. If you looked at all the data, it looked like there was a 25% reduction in mortality, and we thought, God, if anything like that was true, that would be a big step forward. You have to remember, when I went to medical school in the late 70s, we were taught that heart attacks were not due to clot. We were taught they were an electrical instability, and that the clot was secondary to the heart attack. The heart stopped and then the clot formed. And the idea that the clot was actually causal really wasn't thought to be the case. There had been these trials of clot dissolving therapy, and they hadn't shown benefit. They'd actually individually had been used to reinforce the point that the clot wasn't the cause. But if you looked at them together, you came to the opposite conclusion, and the question was which was correct. So we thought we got ISIS-1 was running, and we went and talked to Höchst, who owned the company Boehringer that made streptokinase. And at that time in Germany, there were 2 German cardiologists that were starting to use a streptokinase in acute MI, just trying to test it out with a very different regimen. So rather than over 24 or 48 hours, where you gave the infusion and you had the risk of bleeding for a long time, they had come up with this very short-term regimen, a very high dose. So this was Rolf Schroeder and Professor Neuhaus. And we talked to them, and we thought, well, what about testing this high-dose regimen? First of all, for safety and side effects, and then ideally into a large-scale study. So Höchst agreed to fund a pilot study. They owned the company Boehringer that brewed streptokinase. We went there and visited. It's like going to a brewery. These great big vats, and the bacteria being used to produce the enzyme, which wasn't being used for anything pretty much. A little bit for pulmonary embolism at the time. And they agreed to fund it, a pilot study, and we ran the pilot study of streptokinase in Oxford and in a few other hospitals around the country with mates of mine who we'd been at medical school, and I managed to persuade them to join in. I remember one of the first patients that we gave the treatment to. we randomized to, and they got back pain, and the consultant in charge of them was very unhappy. I was summoned to the ward round. I was told, this is incredibly dangerous stuff. And then I unblinded, and they were in the placebo group. So we were allowed to continue. And the pilot actually showed that there wasn't much in the way of bleeding. There were no obvious symptomatic side effects. And we got approval from Herbst to get funding and the drug to do a large-scale trial of intravenous streptokinase in acute MI. And ISIS-1 finished. Results were not particularly promising in terms of the benefits of intravenous beta blocker, but we had built this network of hospitals around the world. As I say, district general hospitals, the places where most of the MIs are treated, that were interested in going into another study. And we'd also helped to build a network in Italy, and the Italian group, ISIS-1 was going on for a long time. They decided that what they would do is they would stop ISIS-1, and they would run a parallel streptokinase trial based on the meta-analysis that we had done, based on our pilot data, that they would run a parallel streptokinase trial in Italy while we ran the ISIS trial elsewhere. And that's really the formation of the GC group that compared streptokinase versus open control in around 12,000 patients, and then in parallel ISIS-2 elsewhere in 17,000 patients. And GC was the first to publish. What presented, I think, at the American College of Cardiology, and there was disbelief. First of all, it was an open trial. I think there was a bit of prejudice about it having been done in kind of Italy. And there was a reduction in mortality of 20-25%. People thought, nah. And then a year later or so, ISIS-2 came out, placebo-controlled trial, 17,000 patients, absolutely the same result. And the two trials together showed very, very clear reductions in mortality with streptokinase. And I still remember when those results came out. I mean, it was extraordinary. The funny thing was, when we got agreement to do the streptokinase trial, we thought, well, what else? Could we test something else at the same time? And Richard had done a meta-analysis of aspirin post-MI that was the first presentation to the US Food and Drug Administration of a meta-analysis. And on the basis of that, they had approved antiplatelet therapy post-MI, first time. So we thought, well, what about aspirin in the acute phase? You're opening up a coronary artery. If that's what you're doing, getting rid of a clot, would it be useful to have something that prevents the clot from forming? So I think it was over a lunch, actually, at Peter Slight's house one Sunday. We thought, well, why don't we factor in aspirin? Why don't we do a factorial study, streptokinase versus placebo, and effectively randomize again, aspirin versus placebo? Everybody was going, well, first of all, on the contraindication for streptokinase, it said, do not give if someone has had aspirin. So it took a little bit of time to persuade the company. But also people said, aspirin? Aspirin for heart attack? I mean, you're joking, right? In the US, some of the kind of experts were very negative about that. But anyway, we tested it. And as I say, when the result came out, and I saw it for the first time, Richard Doll had been the chair of the Data Monitoring Committee. We had no idea what the results were. And then we were shown the results. Here was this mind-blowing picture. 13% dead with routine care. 10% dead with streptokinase. 10% dead with aspirin. 8% dead with a combination. And I remember this picture coming out, and then I thought, my God, I'm meant to be at the dentist. And I ran, and I arrived at the dentist 15 minutes late, and he put me back in the chair, and I said, yeah, I'm sorry I'm late, but look at this result. And, this kind of graph of this fantastic separation in mortality, that you could nearly halve the risk of death in people presenting out to 12 hours. And if you looked at the people presenting within 4 hours, it was more than 50% reduction in mortality with cheap generic drugs and reinforcing the juicy result with streptokinase. It was an extraordinary moment.
I mean, so many questions from that, but just whilst you're on this thread, what happened next for you and for streptokinase in MI? What happened with the rollout of that?
Well, the British Heart Foundation was incredibly helpful. So while the trial was going on, I contacted Desmond Julian, who was the medical director, and I said it would be really interesting to see what happens before and after a trial result. And so through the British Heart Foundation, we surveyed all the cardiologists in the UK as to whether or not they used fibrinolytic therapy, whether or not they used aspirin in the acute phase of MI. And this was after GISI had been published. And then we did and we got a fantastic response. People did give, we got a very high response rate. And then we did a survey a year after the ISIS-2 results came out. And there was a massive shift from people never using fibrinolytic therapy, very rarely using aspirin, to almost everybody using fibrinolytic therapy and aspirin for at least a large proportion of their patients. And when I think back, why was that the case? I think there are a couple of things. One, the trial was so big that the result was really massively significant. I mean, you didn't have to be a statistician to know that 1,000 deaths versus 800 deaths is real. And I think that was down to Richard's genius. There's no point in trying to persuade a statistician how to treat a patient. You need to persuade doctors. You need results that you don't require sophisticated analysis to make it clear it works or not. It has to be barn door obvious, and it was. The second thing was that the trial wasn't done in the ivory towers. The trial was done in the places where patients are treated by the doctors who treat them. It was their trial. The results were their results. And I think we've seen the same thing happen with the recovery trial in COVID. I mean, Martin Landry, who I work with, says that when he was designing the recovery trial, he had the ISIS-2 protocol open on his desk. He was copying from the ISIS-2 protocol to write the recovery protocol. What kind of data to record at randomization? What kind of data to record at follow-up? How you explain the trial? And the same thing happened with recovery. Recovery was done in pretty much every hospital across the UK. It involved not the consultants, it actually involved the doctors who treat the patients, the more junior doctors who are dealing with the patients as they come in. And so it was their result. And so when dexamethasone was shown to reduce the risk of death in the most serious COVID patients by 30%, it got implemented. And I think it was the same thing. It was barn door obvious, clear-cut result. It was the result that belonged to the people who'd done the trial, not the central coordination team, but the people on the ground. And I think that was what was so beautiful about ISIS, so beautiful about recovery.
Okay, sorry, can you bear me 1 sec?
Was that glass, George?
Yeah. Thank you.
Great. So looking backwards, it seems like such a logical journey to have gone down and such a logical path to have followed. But I'm struck by how you had graduated from medical school and then decided to go off the main path to follow something that is unusual at that time and even today. What drove you to follow your interests and ambitions and trust yourself even when you met those challenges along that way?
Difficult to know. I mean, I don't think I was particularly confident it was going anywhere. I was just kind of interested in it. And I was more interested in and it had emerged over time. I mean, I'd never done statistics. I'd done maths at school, but when I went and did statistics for a year, I really kind of liked it. And when I worked in this trials unit, I really liked it. I liked the fact that one didn't take things on trust. There was a tool that allowed you to determine what worked. And ISIS-2 took what I was taught and showed that it was wrong, that the heart attack was caused by the clot, and that getting rid of the clot and preventing the clot was the way to save lives in heart attack. And of course it's then extended into more effective thrombolytic agents such as TPA, somewhat more bleeding but probably more effective at dissolving the clot, somewhat more effective ways of preventing the clot than aspirin alone, and of course other ways of keeping the artery open. But they actually all flow from GIST and ISIS-2. You need to open the vessel, you need to keep it open. And in fact, even if one looks today, I don't know, but I suspect if we looked at people who get treated for heart attack across the world, it isn't primary angioplasty. I mean, it is if you go to the big cardiology conferences, it's all primary angioplasty and stents. But if you're out in the sticks, you're in rural, Russia, China, across the world where you're not close to the facilities to get primary angioplasty, or you come in too late because of traveling distances, my guess is that thrombolytic therapy, simple antiplatelet therapy is still the most common treatment for acute MI. I'd love to do a proper study of that, but that's my guess. So one gets a little bit of a distorted view from our kind of Western perception, and particularly going to the big cardiology conferences. But be that as it may, I mean, opening up the artery, keeping it open, is still the way we treat MI.
Yeah, I mean, it's such a famous graph of those lines that you refer to of 13% mortality, then 8% mortality. Do you ever think about how many lives are potentially saved in that 5% wedge, and that your work that you were doing in Dr. Yusuf's bedroom has turned into that physical impact?
Sometimes. I mean, I remember we went back to Höchst after we got the results. They invited us back to talk about the study results to their team, to all of their staff. And when Peter Sleight was talking, he was saying to the people, you may think of yourselves as people who sell a drug, but I mean, what I want you to think about now is what you're doing is you're saving lives. This drug saves lives, and that you're not salesmen, you're actually lifesavers. And I think understanding that the pharmaceutical industry is a force for good, working with the pharmaceutical industry to do the right studies, it can be a very positive experience. And those collaborations were, I think, good for us. They were an opportunity to get access to the drug and the placebo, which we wouldn't have had, to funding that we wouldn't have been able to get. But I think also very beneficial for the company. They had a drug that didn't work. No one was using it for anything. In fact, it was a lifesaver. It was a very positive way of kind of putting it, I thought.
Yeah. I'll just ask a question now, to give you a chance to talk about the arson story if you want to. What were the main challenges when implementing a trial of this scale?
Well, the difficulties initially with the ISIS trials was how do you get people interested in joining into such a study. And we were very fortunate in that we linked up with somebody in a pharmaceutical company who was keen to do that, and who had contacts in other parts of their company in different regions that helped to create a network. So I think the first difficulty is to create a network of people who want to do the trials. And the great thing about the ISIS trials was they involved the hospitals that don't normally get involved in research. And I think that was a very positive thing. I think you're learning how you can make a difference. You know, you're not just treating the next patient, but you're part of an enterprise that works out how you should be treating patients. I think it was a very positive experience. I mean, I still meet people who've been involved in the ISIS trials, and it meant something to them. They were actually part of the generation of evidence. So I think getting that going is critical. Often what you're doing in these trials is testing things that people already know the answer to, or so they believe. The recovery trial in COVID, the one treatment that people were very negative about including was dexamethasone. They said it's dangerous to give dexamethasone in such a situation. It's the only one that's really come out with a positive result. The idea that thrombolysis and aspirin would be beneficial was absolutely the antithesis of the teaching at the time. So that's a big obstacle. You know, you're often testing people's very strong prejudices about what does or doesn't work. And you know, the kind of classic trial in that is the CAST trial of antiarrhythmics post-MI. I mean, so sure were they that these antiarrhythmics would be beneficial that they only had a one-sided p-value. So it was either going to not work in terms of reducing mortality or it was going to be beneficial. They weren't testing for harm. And they set it up for benefit. You know, they made sure that the antiarrhythmic was an antiarrhythmic, prevented the arrhythmia in that particular patient. Then they randomized them to the antiarrhythmic versus placebo, and they had to stop the treatment because of harm. The trial because of harm with enconide and flecainide, because their beliefs were so strong that it couldn't be harmful. That they weren't even open to testing that formally. Until they had to stop the trial because it was harmful. So that's another big obstacle. You are testing people's strong prejudices quite a lot of the time, and we've had the same problem when we were looking at cholesterol lowering, that there were very strong prejudices about higher cholesterol was not a problem in older people, that reducing cholesterol below the normal range was hazardous. These were very strong prejudices to the point where it was very difficult to even test them. With thrombolysis in acute MI, based on animal studies, people said even if it did work, it could only work within the first hour or two of a heart attack, because in the pig model, if you gave it later than that, it didn't do anything. But of course, an artificial MI and the real thing, which may be more stuttering, might be different. And it turned out to be the case that even out to 12 hours, although there was a gradation in a smaller risk reduction as time went by, there was still benefit from treating patients with thrombolysis later. But that, even to test that, was considered to be wrong.
Getting centres to buy into the idea of treating something as dramatic as an acute MI is reasonable, and similarly with COVID, it was what we were confronting, and so getting centres to buy in there was reasonable as well. You've done a lot of work on statin therapy, which is much more of a preventative drug, where the disease is not seen in the face as much as it is prevented from happening. How did you develop that idea, or even come to the idea that the idea that lowering cholesterol in patients would have such a dramatic effect?
So back in the 80s, again, there was an enormous controversy about whether or not lowering cholesterol, particularly LDL cholesterol, was beneficial. There had been a number of trials done of lowering cholesterol with diet and with drugs. I think most famously the WHO clofibrate trial that was run by Michael Oliver, relatively small, but the trends in that trial were adverse. So those people who got clofibrate didn't do better than those who got placebo. And so there was a lot of controversy as to whether lowering cholesterol itself would be beneficial. Was it just non-causal? Or was it the drugs that had been used to lower cholesterol? Some of the trials used hormones therapy. Or was it the trials were too small? And again, that's where I think Richard Pito helped to kind of give clarity on that. So he had spent an awful lot of time on creating what I think is perhaps one of the most important never published papers. I think I probably got the only copy of it in a file drawer, in fact, where he got all of the data from all of the cholesterol-lowering trials, randomized trials that have been done, with all the drugs and diet. I mean, some of these trials were done in care homes and in the States, and the individual trials had some weird results, like more trauma deaths in one group than another against treatment, more cancers in the cholesterol-lowering group than the other, in one trial but not in the others. And so he collected all these data together, and again, when one looked at the data, it suggested the trials had been too small and too short. That if you run your cholesterol levels at a particular level for 40, 50, 60 years, it's pretty optimistic to hope that within a couple of years you'll reverse that. So there were two things that were important. One was to get the epidemiology right. One was to understand what is the association between cholesterol and coronary artery disease. How strong is it? And then to consider the randomized trials in the context of the epidemiology. So you run your cholesterol levels high for 40, 50 years. You lower your cholesterol for a few years. You can't expect to get a bigger reduction in risk than is observed in the observational epidemiology. You can't reverse all the risk, or more than all the risk associated with that slightly higher cholesterol. And yet that's what the trials really had assumed. They hadn't been designed thinking about the strength of the relationship between cholesterol over a lifetime and coronary artery disease. They were looking for a bigger than epidemiological expected reduction in risk by just lowering it for a couple of years. So if you looked at the trials in that context, you realize the trials were too small to pick up even a full reversal of risk from lowering cholesterol for a few years, let alone some proportion of that. And what the data suggested was that you needed to lower cholesterol for longer than just a couple of years, that the trials of 2 years versus, say, 4 years on average had a smaller benefit. The more you lowered the cholesterol, the more you lowered the risk. But none of the trials on their own had been big enough. So that provided a context. And then we were invited to go to China, initially to help with a randomized trial that was ongoing of hepatitis B vaccination on cancer. But at the same time, we went to, this was in the kind of mid-80s, we went to the first tobacco conference in Tianjin. Took the train from Beijing up to Tianjin to this conference. And again, I remember we were in the coffee break, and this small Chinese lady came over who was a professor in Shanghai. And she told us this extraordinary story and introduced us to her PhD student, that in the Cultural Revolution, she had been forced out of her job, and she was doing menial work, things like toilet cleaning and stuff like that. And so at the same time, she managed, I don't know quite how, to measure the cholesterol levels of several thousand people in various factories in Shanghai, I guess through kind of contacts and whatever, just to get a kind of idea about cholesterol level in China. And again, I don't know what really drove that. And her PhD student, many years later, one of the first medical students post-Cultural Revolution, Zhengming Chen, was doing his PhD by taking this cholesterol data from a decade or so before and linking it to the health of the people who were in that study. And what was really exciting to Richard and I was that the cholesterol levels were way below the Western range. So in looking at the randomized trials, studies like by Ansel Keys in America had compared cholesterol in different populations around the world with their heart disease rates and showed populations with lower cholesterol had lower risk. But was that because there were differences in those people other than cholesterol? Lower risk of coronary disease. Studies within populations, perhaps the most robust was the Mr. Fit screening study in the US in middle-aged American men, and about a third of a million of them showed a log linear association between cholesterol and coronary artery disease within the Western range, suggesting lower cholesterol, lower risk. But here was this extraordinary data of cholesterol levels that were way below any levels one could really study properly in the West. And still there was this log linear association, lower cholesterol, way below our normal range, lower risk. So that really kind of made us think, well, if you had a drug that could lower cholesterol much more than the previous drugs or diet, and you took people who are at high risk because they already had vascular disease, then lowering their cholesterol, irrespective of their cholesterol level, should lower their risk. Because it's a log linear association in the epidemiology, where each 1 millimole difference in cholesterol is associated with a 50% difference in risk. Let's say you get half of that. You would anticipate, within a few years, you would anticipate each 1 millimole reduction in LDL cholesterol would produce, let's say half, a 25% reduction in risk. 2 millimole reductions, 2 quarters, multiplicative, so about 40% reduction in risk. So the more you can lower cholesterol, the better. The higher your absolute risk, the better. But it doesn't matter where your cholesterol starts in terms of how much risk reduction you get per millimole. Now, that was really kind of based on this study that was established by somebody who was working in cleaning toilets. And we were talking in this coffee break to Zhengming Chen, and his professor was saying the problem is we don't have any decent computing. He's really struggling to do this. And we said, well, would he like to transfer to Oxford and do his PhD here? We'd never had any PhD students. We were a very small group. And she said, oh, that would be fabulous. So that's what happened. Zhengming Chen came and did his PhD here. And then subsequently has set up a big cohort of 500,000 people in China, the Cardiology Biobank, which he continues to work on. But it was that observation that maybe there wasn't a threshold. Again, completely against the perceived view. All the guidelines that came out on lowering cholesterol, you said you mustn't lower low cholesterol. That would be not beneficial and potentially hazardous. Similarly, when people had done observational epidemiology, on small-scale studies, they come to the conclusion that higher cholesterol was not associated with higher risk in elderly people. But again, when we combine data from observational studies, it looked to us to be pretty much log linear at all ages. So we were really keen to look at lowering cholesterol in high-risk individuals, irrespective of their age. Older people are obviously at much higher risk, so it's important to know whether one can produce benefit in them, irrespective of their cholesterol level. And then the statins emerged. So we were talking with Merck as the statins became a possibility when they were doing their early studies. We did a pilot study here in Oxford. Jane Armitage, who I still work with, ran the pilot study here in Oxford, looking at potential side effects of lowering cholesterol with a statin. I mean, it's a completely new drug. It was a drug that was nearly killed by some adverse results seen in beagle dogs, I think, where Merck closed everything down for a year while they decided what to do. They did agree to then move forward into clinical studies. We did pilot studies looking at the effect on mood, on sleep. There had been earlier drugs of cholesterol lowering that had adverse effects on cataracts and things like that. So we did very detailed eye studies working with Tony Braun and his colleagues here in the eye hospital. No side effects. So then we went to the British Heart Foundation, the MRC, and talked with Merck about doing a large-scale cholesterol lowering study, 20,000 patients we wanted to test, high-risk individuals, people with previous cardiovascular disease or diabetes, irrespective of age, irrespective of cholesterol. And you ask about obstacles, that's when we hit obstacles. We just couldn't get it funded. Merck had moved on, so we couldn't get funding from their research side. The MRC advisors were saying dangerous to lower cholesterol in people who are old or in people with below average cholesterols. And there were a couple of things that made the difference. The British Heart Foundation. The British Heart Foundation were the first to say, if we will put £1 million down on the table if you can get the rest of the money. So the BHF kind of made it seem more credible, more acceptable. That then helped to get some others to at least listen. And the Merck subsidiary in the UK said, this is exciting. This could be something, we could put the UK Merck on the map. So the head of Merck UK said, I'll match that $1 million. So now we were at 2 million. We were looking for about 20 million for this trial of 20,000 patients over 5 years. We kept on talking to the Merck research team, but not interested. And then somehow we got in touch with the Merck marketing people. And the guy who was in charge of marketing for South America. He got interested. He thought maybe this trial, by just demonstrating, I guess, safety and efficacy, would be useful in convincing people to use the drug. He was interested. And then, I mean, this just gives a sense, I think, of how random stuff is. Richard went to a meeting in Spain with a Roche meeting, I think he was talking at. And he started talking with them, and they were talking about doing a trial of their vitamin cocktail. And Richard had done a lot of work with vitamins. In fact, when I met him when he was fixing a chair, the one paper he gave me was a paper that he and Richard Doll had written on beta-carotene and cancer. So I was sitting reading that while he was finishing off his conversation with Richard Doll. So he had done a lot on vitamins, and that's why he was at this meeting with Roche. And he said, well, why don't you factor it into this cholesterol trial? And so Roche said, okay, how much would it cost? So they came up with 1/3 of the money. So now we were kind of piecing this together, and we went back to the MRC, and they rejected it again. And we went to Merck and the marketing people, and they said, well, if you get the MRC in, we're in. So we went back to the MRC again, and there was an officer in the MRC who thought, this is weird. What's going on here? And for whatever reason, Tim Sprosen, got out all the files and really looked at it all. And then he went to his senior colleagues and said, I think we're making a mistake. I mean, I'm surmising that this is what happened, because we were then invited back for another meeting to present this. And the MRC decided to fund it. I mean, Richard Doll was so surprised by this, by after 3 rejections getting it funded, that he bought champagne for the group. He said, I've never seen anything like this happen. By that time, Richard Pito was completely exhausted by the difficulty of getting it funded. So I was sitting with Jane, and we had this money, we had the drug, and we had the network of UK hospitals that had worked in the ISIS trials. And so we were ready to go. And that was how the heart protection study happened. The MRC had only 3 conditions. They wanted to change the name of the trial. I said, you can call it whatever you want. You can't include people aged over 65 because of the concerns. And you need a lower cutoff of cholesterol. So Jane and I looked at the data and we came up with a threshold of cholesterol that would exclude no one. And we told them that threshold and they said that's fine. And we couldn't get them to budge on the age. Until about 6 months later, I got a call from another part of the MRC head office saying, we have a policy about not excluding people from trials on the basis of age unless there's a good reason. Why are you doing that? And I said, I don't know, there isn't a good reason. They said, well, you can't exclude them on the basis of age. So the MRC got rid of their own precondition. And then we included people of all ages and effectively down to any cholesterol level. And after 5 years of follow-up, these amazing results emerged. Reductions in coronary artery disease, irrespective of your starting cholesterol level, a reduction of about 20-25% for each 1 millimole reduction in LDL cholesterol, in males and females, in young and old, in people with or without diabetes, and incredibly well tolerated and safe. I mean, really no difference between the active and placebo group in terms of any side effects. So it built on some of the other studies that had already come out, the 4S study of high cholesterol in people with coronary disease that had shown, the first trial that showed benefit with statins. But it was really the one that showed lowering cholesterol at low levels, lowered the risk in diabetes. There'd been an unexpected finding in another trial that had just come out, the lipid trial in Australia with pravastatin, of a possible reduction in stroke, which was unexpected because the observational epidemiology doesn't show such strong associations of LDL cholesterol with stroke, even if you look at ischemic stroke. But we got a much stronger confirmatory signal on stroke than in lipid. So again, demonstrating benefits on ischemic stroke and no effect on hemorrhagic stroke. So that was a pretty exciting moment, I have to say.
Yeah. I mean, your approach to trial design sounds relatively simple in the sense that I think in the statin studies you didn't measure cholesterol at every attendance, and that's almost counterintuitive and leads away from any sub-analyses and things like that, which are commonplace today. Was that out of necessity because of the funding issues you mentioned, or was that what was the driver behind keeping things that basic, say?
Well, I think the thing about clinical trials is there's a number of elements to it. I mean, it's not that hard to come up with a good question. The most interesting part, though, is designing the trial to answer the question. And so with respect to answering the question about whether cholesterol lowering lowers the risk of coronary artery disease and death, you're not interested in the cholesterol levels. What you're interested in is how many people have a heart attack, how many people have a stroke, how many people die. You already know the drug lowers cholesterol. You've done that previously, that's why you're testing it. And so in many respects you don't need to know the cholesterol levels once you've randomized the patients. The drug lowers cholesterol. All you're doing is essentially a test of compliance with a measure of cholesterol. So it's true, it is counterintuitive until you actually work out what you're trying to do. And so almost all of the other statin trials, every time the patient came in, they collected a blood sample and they measured total cholesterol. And I thought, well, why would we do that? What are we trying to actually get at? And what we're trying to get at is, what is the cholesterol difference that we see in the trial as a whole? And so to get a difference in 20,000 patients, you don't need to test 20,000 people. You only need to test a random sample. And so I thought, well, we just need a random sample of about 5%. 1,000 people will be adequate to determine what is the average cholesterol difference in the trial, which is what we want to know in terms of, well, what risk reduction do you see with what average cholesterol reduction? And of course, if you're testing only 5%, if you're only testing 1,000 people rather than 20,000 people, and you're only doing it once a year rather than every visit, you can test much more. You can actually analyze not just total cholesterol, which is pretty much what all the other trials did, but you can afford to do the apolipoproteins, the LDL, the HDL, direct LDL, none of which was done in the other trials because, of course, they're doing it in every patient, every visit. They can't afford to do more detailed assays. But we could do more detailed assays at lower cost because we're only doing in a small proportion. And therefore, we're able to say what are the average differences in all of the components of lipids at lower cost. So we get more quality, more data at the right statistical precision. We collected the samples at baseline because you want to characterize the participants in terms of what is their cholesterol level. Again, their apolipoproteins, their LDL, their HDL at baseline. But when you do analyses of post-randomization lipid levels, one of the major errors is to think you can compare the people who get a big reduction in LDL cholesterol post-randomization and those who get a small reduction, because it's no longer a randomized comparison. The post-randomization cholesterol levels that can't be applied to the risk reduction because you're comparing the people who get treatment with other people who get treatment. You're ignoring the placebo group, so you're essentially ignoring the randomization. So all of this data that people get that they then use to try to determine, do you get a bigger reduction in risk or a smaller reduction in risk among people in the treated group who get a bigger or smaller reduction in cholesterol, are non-randomized, confounded evaluations, and therefore really go against the whole principle of a randomized trial. And you think, well, why do you do a randomized trial if you think you determine the effects of treatment in a non-randomized way? That to me is really counterintuitive. Yeah. And I mean, there are other ways in which I think one can spend a lot of time thinking about the design that can produce improvements. In ISIS-1, we asked the doctors to complete a very simple form in the atenolol trial and send us a copy of the pre-randomization ECG so we could analyze them. And I analyzed 17,000 ECGs, measuring them. And I thought, well, first of all, sometimes I didn't get the ECG. Secondly, sometimes I got an ECG and I wasn't sure it was pre-randomization or whether it was after the treatment where it might have been influenced. So I thought, there must be a better way. And I spent many hours analyzing these ECGs, as you can imagine, recording all these measurements. And then when I looked at the paper for ISIS-1, this had all been distilled down to 6 groups saying anterior, inferior, etc. So I thought, hey, I'll just ask that question at randomization. The doctor's got the ECG. I know it's pre-randomization because he's not yet randomized the patient. I'll ask him, which group are they in? He's got the ECG, or she's got the ECG. They tell me, I know it's pre-randomization, and I don't have to chase up ECGs. No one has to analyze them, and it's better data. So I think there are a lot of things that one can do in randomized trials which are quite fun. Factorial trials, testing aspirin on top of streptokinase, testing a cocktail of vitamins while testing statins. 2 trials for the price of 1. We found the vitamins didn't work. But these are things that are often not done. And you have to ask the question, why not? And I think a lot of it is just not spending enough time thinking about the design. The question's easy. The design is where the fun is, and then the delivery.
On that point, your work has shown some absolute risk reductions in hard endpoints, and today you could argue that a lot of the big gains we're going to make in cardiovascular endpoints and survival have been achieved. So how do you think people running trials today need to adapt their work to try and produce studies of value for practice today?
Actually, I mean, maybe I'm a natural optimist, but and to some extent in research you have to be. One puts in research applications with the expectation they will be rejected. So optimism is kind of a key characteristic, I suspect. But I think that this is a fantastic time to be doing randomized trials. The obstacles that have been created in the last 20 years are, if you like, of our own making. They derive from over-regulation and bureaucratic obstacles to doing randomized controlled trials. We wrote a paper in, I think it was published in the New England Journal in February 2020, called the Magic of Randomization versus the Myth of Real-World Evidence. And the argument in that, it just predated the pandemic, the argument in that was that because it had become so difficult to do randomized trials, driven by the drafting of the International Committee on Harmonization's Good Clinical Practice in 1995, a whole set of very poorly considered rules and regulations, over-interpreted, the creation of the CRO industry, didn't exist in 1995, now a $70 billion a year industry, layering on more and more obstacles to doing trials. People have been pushed away from them to trying to use so-called real-world evidence, observational, non-randomized data. And I think it's a false friend. With observational studies, you always have the potential for confounding. You can never be sure that you're comparing like with like. The magic, if you like, of randomization is if you randomize large enough numbers of people, the two groups will differ from each other only randomly by the play of chance. So you're comparing like with like. And then if you see a difference in outcome in one group versus the other that gets a treatment, you know it's due to the treatment, not due to some kinds of confounding. And we've seen that in a lot of the nonsense around side effects associated with statins. Nothing seen in the randomized trials, all sorts of claims based on observational data which are completely confounded. So I think the emphasis on observational real-world evidence is very, very damaging. I think it's been driven by the very poorly written guidelines that came out of the regulators that were then over-interpreted to the advantage of the creation of the contract research organizations. I think there's now an opportunity to reverse that trend. We've seen what can be done in the last 2 years if you actually start thinking. The recovery trial is an example of that, but the vaccine trials as well. One can randomize at large scale, one can demonstrate benefits that wouldn't otherwise emerge. And we have the ability to do this so much more easily than in the past. With the data systems, with computerization, we can identify people through health record systems, invite the right people into trials, do it in a very much more efficient way. We can follow up patients through their health record systems again, very much more efficient. You can reduce the drudge for the clinician in these trials, but actually get a lot of really important questions answered. So I am actually quite optimistic. I think we need to kind of push back and make it possible to do large-scale trials again. The last 20 years have been very, very damaging. What we've seen with industry is because of the increasing costs of trials, they've shifted away from large-scale, long-term trials in chronic conditions like cardiovascular disease, moved into small niche treatments where they charge a lot of money. But from a population health perspective, very, very adverse. A nice piece of work done by Clive Meanwell in the States showed that over the last 10 years, sales of drugs had gone up by 2.5-folds in the top 10 drugs, but the population covered by it had reduced about 7-fold. So from a population perspective, that's really bad news. I think that has to be reversed. And it's not, there's not a shortage of patients. The question is how to get them into clinical trials, how to make it easy for them to be in the trial, and how do we make it easy for the clinicians. And I think the key thing to that is reinvent randomized trials for the 21st century effectively.
Yeah. From the examples you gave, the work you did seems to be conceived by yourself and physicians and then taken to industry because you needed the tools that they can provide for support. And it seems that today that relationship has almost flipped a little bit, where industry provide the tools and then want the support of physicians to provide it. What would you say to fellows or physicians who have ideas and want to try and push them forwards when it seems like the energy is coming from the other way?
Well, I think there has been a big shift over the last 20 or 30 years to the companies working much more closely with CROs, because the contract research organizations promise to follow all the rules and more, because the more they do, the more they charge, the more money they make. And so there has been an escalation in this and a move away from what I think was a very, very creative collaboration between industry and academia, which is one that we need to recreate. We've built up relationships ourselves with the people in the companies, and I think one builds trust about doing trials that do get regulatory approval, but doing them in a smarter way. I think also it's worth understanding where the new drugs are coming from. So the new drugs are largely being developed in these kind of small biotechs, and they're in a bind because they don't have the money to do a very large scale trial in the way it's done currently. And therefore there are a lot of really interesting drugs that are being created by these small biotechs. They have to find someone with a lot of money, one of the big pharma, who will essentially take it away from them and take it to the next stage. And it's a little bit like the lottery. If you want to win the lottery, you have to buy a lot of tickets. And we've made the ticket price so high that people can only afford to buy a small number of tickets. So you have a lot of really innovative treatments, but only a small number of them will move forward. And I think one of the best examples of that in cardiology is PCSK9. So PCSK9 inhibitors. I mean, PCSK9 are kind of the poster boy in cardiovascular disease or poster girl of genetics. Finding a target through genetic work, that this protein was important in the recycling of LDL receptors. If you have genetically no PCSK9 or almost no PCSK9, you have much lower cholesterol levels. If you have intermediate levels of PCSK9, you have intermediate levels of cholesterol. And within 20 years, not only developing antibodies to the protein and doing the early studies, but actually doing large-scale mortality studies is fantastically fast. But those trials were so expensive that the companies that did them, Amgen and Sanofi, did big trials, but they did them for too short a time. So had a repeat of the 1970s of cholesterol lowering, of lowering cholesterol quite a lot, but not lowering it for long enough. So the trials showed reductions in coronary disease, but not as much as you'd expect, because the first year you don't see any benefit. The second year you see benefit, but you need longer to see all of the benefit. No benefit on cardiovascular disease because that takes cardiovascular death because that takes longer to emerge. And so I think the Amgen trial is reputed to have cost $2 billion. And the result was considered by the markets to be a dud. So it pretty much killed the whole idea of PCSK9. Now, this very, very smart chemist, John Maraganore, had come up with an alternative way of reducing PCSK9 levels. Rather than mopping it up with an antibody, which you have to give as an injection subcutaneously every couple of weeks, he'd worked out how to block the synthesis of PCSK9 with an injection you give every 6 or 12 months. This had been picked up by Clive Meanwell, who had set up the medicines company, a Birmingham medical student alumnus, and they were in this bind. The market said PCSK9 inhibition wasn't effective, couldn't sell it. So no one wanted to take over inclisiran and do a large-scale trial. So we started talking with Clive, and I'd known him and John Maraganore for a long, long time, and persuaded them that the way to do it was to do it smart, to do a very streamlined, much lower cost trial as a collaboration between the company and academia. So with the TIMI group in the US and with our group in Oxford, we jointly established this trial of high-risk patients, 15,000 high-risk patients of inclisiran versus placebo, which is now going on. He couldn't get any of the companies interested. The moment that trial was started, and it was clear that one would be able to get evidence that inclisiran was beneficial at a reasonable cost, then that drug became valuable. And Novartis bought the company for something like $9 billion. But it was an example of what's happening all the time now, that there are these really interesting biotechs creating these fantastic new drugs. But the obstacle to doing the large-scale trial is the money, not the lack of patients, not the lack of a need for further beneficial treatments. And I think drugs like inclisiran could be completely transformational, because we now have genetic data like the polygenic risk scores that show how you can identify people at relatively young age who have risk based on a combination of genetic factors that is equivalent to having FH, 3 or 4, 5%. You're probably not going to want to take a statin when you're 30 for the rest of your life. But the idea you have an injection once a year, vaccination against coronary artery disease, hey, why not? So we do need these additional treatments, either instead of, because they might be more attractive in particular circumstances, or as an addition. So when you're really at high risk, the combination of an injection every 6 months or a year, plus taking your statin, again, why not? But the question is not have we solved it all and the risks are so low. The question is, how do we solve the problems we've caused to being able to do randomized trials? And they are regulatory. Actually, the practical aspects of doing trials much easier now, because the 21st century is different from the 20th century. We have an IT capacity, a data capacity that could transform the way we evaluate everything. We could have randomized in COVID when you give the second dose of the vaccine, rather than arbitrarily changing it from 4 weeks to 12 weeks. It would have been easy to just randomize through the appointment system when people were invited back. We could randomize whether or not you give a booster. All of this stuff is really easy to do. The obstacles are in here. The obstacles are in people's minds. And that's the thing one has to overcome most of the time.
Great. Pivoting now, you're also the principal investigator of the Biobank UK, which has been the foundation for so much great research in cardiology, but across medicine as well.
Sorry, can I bear you 1 sec? We've got 10 minutes of card on that camera, and we've got 2 very nasty light sources coming in behind, just to let you know. So I can change card on here. In about 15 minutes, it's going to start hitting support, or he's.
I think we can close up by in
Just
15
to let you know,
minutes.
it's just, yeah, the final moment is just to let you know.
And do you know whose phone that is? It's buzzing. Is that a phone or mine? Seems to be.
No, not mine.
Okay. Is it my phone? Shouldn't be buzzing. So,
Oh, is that on there? Oh, I didn't go on.
just double check.
The battery's fine.
Let me just check it.
Sure.
Yeah, it's mine. Sorry. No worries. Cool. Do you mind if I
Actually,
move across?
Yeah, I'm just going to put everyone set. Just coming out for a sec.
Starting.
Yeah, you keep rolling. Yeah, I'm still rolling.
Should we just can't do anything or should
Great. Are we waiting? Is he swapping the card?
He's just swapping the card around.
Okay.
The camera.
I think just the last bits will be, I'll just ask you about what you hope to achieve at Biobank, and then looking at the future of RCTs with the modern technologies that you've discussed and computer modelling, and whether that can all support and genetic testing and things as well.
It's really good though. It's fascinating. Thank you. Yeah.
Okay, I'm still rolling, by the way.
Is it the right kind of length, or is it too long?
We'll be able to cut anything
Okay.
and sort of mash it all together. Okay. So you're also the principal investigator of the Biobank UK, and that's led to so much great results in research in cardiovascular medicine, but also across the other specialties. What are your personal aims in that role, and what do you hope to achieve?
Well, being involved with UK Biobank has been an enormous amount of fun. I was on holiday in Italy actually, and I got a call from John Bell, the Regis Professor here, who had been involved in, I think, persuading the MRC and the Wellcome Trust to fund a large prospective study. Richard Peto and I had been trying to get funding for a very large prospective study where one would be able to look at the association of many different kinds of risk factors to disease. We'd spent a lot of time combining data from small prospective studies of cholesterol, of blood pressure, to try to get a sense of the associations between those risk factors and disease, so taking old studies. And we realized, as with randomized trials, that you needed to have very large observational studies in order to be able to get really clear pictures of the associations of risk factors with disease, particularly if you wanted to look at the combination of different risk factors, say blood pressure and age, and what's the slope, what's the shape of that association, what's the strength of that association. And so we felt there was a real need to set up some large-scale prospective studies. There are other people, Nick Day in Cambridge also of that view. And we had been successful in getting funding from a Hong Kong charity, from the Kadoorie family, to set up a study in China. So my colleague Zhengming Chen, who had finished his PhD and was now working, set up the China Kadoorie Biobank in China, identifying 5 rural and 5 urban areas where they had very good disease surveillance, so we'd be able to follow up the health of the people. And with that funding from the Kadoorie Family Trust, recruited 500,000 people in China, or started to do that. And then at the same time, the MRC and the Wellcome Trust, at the beginning of the century, had got interested in the idea of funding such a study, and you decided on a couple of things: 500,000 people, people aged 40 to 69, and that was about it. And then they set it up as a charity. They got a team in place, but the question was, how do you deliver it? And the whole thing was struggling a bit in around the delivery. And so I got this call from John Bell saying they were looking for someone who might be able to help deliver it. And because we had done randomized trials like Heart Protection Study, where we'd used health record systems to identify potentially eligible people and then invite them centrally into the clinics and run all the appointments. I thought this model could work for recruiting 500,000 people in the UK. We could get their names and addresses for people aged 40 to 69 from the NHS. We could write to them. We could invite them to special clinics we set up. And Zhengming Chen in China had started doing the study, and he'd come up with this idea of essentially what it was like a little factory production line, where a person came in, they were with one staff member for a while doing one bit, then they moved to the next station, then to the next station, next station. So people were moving through this production line. I thought, well, you know, that could work as well. One could have a sort of production line. One could set up a kind of mega clinic in an area. You could invite all the people who are potentially eligible who live near that to come along. You could run an appointment system. I thought, oh, this is potentially doable. So anyway, I was interviewed and I was appointed to take this on, and I was working with fantastically effective guy called Tim Peakman, who was the chief operating officer, and it was all set up in Manchester. And our IT team, who had developed the systems for our clinical trials in terms of recruitment, but also collection of data in the clinic, designed the system for inviting the participants, running appointments in these mega clinics, tracking them as they went through in terms of their data collection, so they all went round with a memory key and they stuck it into the next station, and then put in place a whole series of monitoring systems on the data. So real time, as the data came in each day, they would be running a whole series of checks to see whether there are any kind of anomalies coming up, any problems with any bits of the process. Were there blockages in people moving from one area to another? Were we bringing in too many people and causing blockages? We were running an airline, essentially. And kind of that was a sort of fun thing. It was a process of how do you get 500,000 people in? How do you get the maximum amount of data? And again, a lot of the obstacles are people's preconceptions about what you can and can't do. So in designing the study, there were lots of expert groups coming up with what questions to ask, what measurements to make. And so, you know, the people who are interested in diet wanted 100 diet questions. The people who are interested in mental health wanted 100 mental health questions. And, you know, and they were going to have a nurse or a staff member asking these patients hundreds and hundreds and hundreds of questions. Yeah. It would have taken forever. So one of our IT team leads said, well, why don't we just put it on a touchscreen? And again, now you think, well, pretty obvious, but we're talking 2004, 2005. And everybody went, no, elderly people won't be able to deal with a touchscreen with a computer. No, it won't work. So we went out and bought some touchscreens, dragged some people in and said, can you ask these questions on it? And they did it much quicker. They liked it much more than being asked the questions. They were willing to answer quite invasive questions that they wouldn't perhaps been happy to say out loud. And so, you know, we showed these data and people were like, oh, okay. And then there was the question about, well, we'll have to collect the samples fasted. And we said, really, why? That's going to be quite problematic. So then we did a randomized trial of asking people to come in fasted and asking people, and not asking people to fast. And we found there was no difference in the time since the last meal among those who were asked to fast versus those who weren't asked to fast. So it had no impact at all. Apart from, in the ones we asked to fast, we said, unless you're a diabetic, don't fast. And we found that diabetics didn't read that bit and did fast. So again, evaluating stuff was really very helpful in streamlining it. Design, more thought at the beginning is really worthwhile. There's always the pressure to let's get started. Everybody wants to get started, the first patient in the trial, the whole thing running. But actually spending time thinking, testing, piloting is an incredibly good investment in the long run. And so that was how I got involved in it. It was about the fact that we had experience in doing large-scale studies, and this was just kind of a step up. And we had a clear vision, I think, of how to do it, and it worked. We were able to get much more data than we would otherwise have been able to get because we used the touch screens. We got really high quality data because we used the IT systems to monitor the whole process. It was incredibly efficient. 100 people a day, every day, everything runs smoothly. 90 people a day, we ran out of money. 110 people a day, the system broke and there were blockages. So there was all this fine tuning going on, but we recruited 500,000 people between 2006 2010, on time, on budget. And then we've been linking them into the health record systems and making the data available. And what's been really enjoyable, particularly once funding was made available to genotype the whole cohort and people really got interested in using it, and it became more mature as more and more people had developed health outcomes so you could study them, is just sitting back and watching. Our job is to build it and make it available, but more and more people now using it. I think there are 30,000 researchers using UK Biobank. In 2021 alone, 1,699 papers based on UK Biobank. The publication numbers are on an exponential curve. It's very, very exciting, and in many ways it's one of the most exciting things I've been involved in, because you kind of get a sense of what you can enable, and also you kind of unleash the imagination of people who would never normally get access to data like this, bringing in experts from outside of the medical area that have skills that you, rather than working out how to sell more dresses on Amazon, are working out how to save more lives. I think that enabling power of Biobank has been an incredibly exciting thing to be part of.
Yeah, it sounds like through your career, if I could say, that the main thing that you've been very good at is problem solving, and you aren't willing to accept that this is the way it's done. You think, why is it done like this, and how can we do it better? From the start of the trials until this role now. And more than that, it seems like you enjoy that process. Do you think that's something that you're aware of, or am I off track there, or do you think that's something that's important for researchers?
Well, I think I was really lucky, and particularly lucky to meet Richard Peto. I think Richard always questioned things. And I often felt I learnt more medicine in my first few years working with Richard than I did at medical school, because he was always questioning, what's the evidence? And I think that got you to ask, not only what's the evidence in terms of the treatment, but what's the evidence that it won't work? And how can you make it kind of work better? What are the things that could be quite different that would work better, rather than just accepting the status quo? So I think that I kind of picked that up from Richard. And I think one of the things that, when I say lucky, is in research, particularly the kind of the early years, and the early years go on for quite a remarkably long time. I mean, I started in Oxford in 1981. The ISIS-2 result was 1988 it was published. That's a long time with essentially nothing to show for it. So you have to be lucky in having someone who believes in you more than you believe in yourself. I think that's more than a mentor. I think of it kind of more like a sponsor. And I was lucky Richard kind of, for whatever reason, believed in me. And I think that's the thing that, as you get to the position where you can help support people, it's more than just giving them a bit of advice. It's actually giving them the chance to demonstrate what they can do, and finding out where it is they can do it. In some areas, you may not be a lab researcher. that may not be your strength. You may kind of start out in a lab and it just doesn't work, but you may be a fantastic researcher in some other area. So it's finding the right hole for your kind of skills, and having enough time to do that and having the support to do that. And I think that's why it's really important for funding agencies like the British Heart Foundation, particularly to support early career researchers. It's one of the things that we've done here in Oxford with the BHF Centre for Research Excellence. I think our best investment has been in transitional fellowships of people who just needed another few years to get to the point where things started to take off. So it's part of what motivates me now, is thinking about what we can do there. The first thing we did when we created the Department of Population Health in 2013, bringing together various groups in cancer epidemiology, cardiovascular epidemiology and trials, perinatal medicine, ethics and economics, was to set up a master's program and a DPhil program to help people and support people to get to the point where they could do research, give them the skills. And now with the initiation from the European Society of Cardiology, some grants from them and from the department, we set up a remote Masters in Clinical Trials, which has been running for the last few years and has a different set of people. So rather than people who are kind of doing their first postgraduate degree, these are often people who are quite advanced in their clinical careers but want to learn how to do clinical trials. And so that's been fun as well, because they have a lot of clinical expertise and some really interesting questions, because they're coming from all over the world. And doing it remotely has meant it's possible for them at that stage in their career to kind of be part of a learning experience. And I think the learning goes both ways, because you are working with people who have really good ideas about what needs to be tested, and what you're doing is helping to give them the skills to do that.
You mentioned that Richard Peto trusted you to wait all those years for your results to pay off and produce. When you're now in the position to be a potential sponsor for someone, what do you look for in terms of personal characteristics in that individual for you to have that faith that they're going to deliver, or it's going to be a worthwhile way for you to use your time?
I wish I knew how to answer the question. I don't know. I honestly don't know. I mean, I've been surprised a lot by the fact that if you can find the right area for a person, someone who struggles in one area can really benefit in another. So I guess the key thing is not to prejudge. Try a few things, support people, give them enough time. I mean, the key thing is people wanting to do it. Like everything, like clinical work, like research, you can't half do it. You've got to really want to do it. So I think that's a big start. If people really want to do it, believe in it. I think that's a critical part of it. But that's true of pretty much everything.
Okay. And from your vantage point in research and clinical trials, looking forwards now, what do you think are the areas that you're most optimistic about, where you think we're really going to drive forward research and change in randomized clinical trials as one aspect, but I'm talking about the advances we're making in technology and genetic studies and computer modelling. How do you think that's going to influence research going forwards?
Well, I'm certainly no geneticist, although as one of my relatively few PhD students, John Jeunesse, said when being interviewed as an epidemiologist, he was asked the question, what do you think about genetics? And he said it's a great way of getting epidemiology funded. And so at that point I did realise that it was necessary to get a defensive knowledge of genetics, and went off and did a few courses, and even spent 3 months in a laboratory at the Rockefeller, trying not to break too many things. I certainly found the niche where I was not suited. But I think the genetics and the genetic data that are coming out of UK Biobank and now other studies are going to be valuable in a number of ways. The most obvious, of course, is identifying new targets for treatment. So I suspect the PCSK9 example is one of what we'll see a kind of burgeoning number of new targets for a range of therapies, and therefore an increasing need for randomized trials to evaluate them if they're not just to get lost. I think that what we're also seeing, the most obvious thing again, is the polygenic risk score, which goes completely against all of the theories. Everybody was writing that there's no possibility that these variants of small effect could be predictive of risk. A PhD student, Amit Kira, working with Setkathiresan at the Broad, his PhD was on essentially the discovery of the value of polygenic risk scores in cardiovascular disease, showing that the combination of many different variants of small effect could actually identify 10 times the number of people that have FH with equivalent risk of cardiovascular disease. I think that finding, which has then been extended to many other conditions, will transform the way in which we roll out treatment. I mean, when you think about the use of statins, for example, or the use of blood pressure lowering therapy, the risk scores are largely driven by age, a bit by the risk factor level, but largely by age. So, you get to 65 as a man, and your GP puts stuff into a score, but essentially all they need to do is look at your date of birth. I mean, it kind of goes up and down a bit depending on other things, but not a lot. It's largely driven by age. Well, if you can use a polygenic risk score and identify someone at 30 who's 3 or 4 or 5% of the population who are equivalent risk to FH, and then you have treatments that can be given like inclisiran as an injection every year, that's a transformation in how we think about cardioprotection. But similarly, screening, breast cancer screening, age 50, colorectal cancer screening, with a really useless test, fecal occult blood, at a particular age. Well, again, if you could use a polygenic risk score, you would say, well, these are the women we should be screening at 30. These are the women we should never screen, because they're the other extreme. These are the people that we should do something more than fecal occult blood. We should actually use a more expensive, targeted screening tool to see whether they have anything that's likely to put them at risk. So I think I can see a number of areas. I'm sure there are many other areas, but where genetics will transform the way in which we have preventive strategies, how we roll them out, and also where we identify a lot of new interesting treatments. And then the question is, how do they get tested properly? And the way not to do it is with observational studies. That is absolutely not the way. So it's absolutely essential that we change the mindset on how we do randomized trials. Otherwise, we will miss an incredible opportunity to take advantage of all of the new treatments that will come out of the genetics, out of things like CRISPR technology, people like Setkathiresan at Verve Therapeutics doing this work where you can actually turn off PCSK9 with a single injection for life. This stuff needs to be evaluated properly, both for efficacy and for safety, and that requires large-scale randomized trials. They can't cost $2 billion a pop, so we've got to use our intelligence. We've got to think, let's assume randomized trials were invented today. Let's forget all the rules. Let's think, how would we do them now? So suddenly I've explained to you a randomized trial, and you think, well, that's an interesting idea. How would we do that? Well, we would use the data to identify the people and to follow them up. We wouldn't be writing stuff on pieces of paper. We would be involving people who are not coming to hospital. We would be, following them up in their homes. Yeah. So I think that there is a reimagining of the evaluation of therapy in randomized trials that's required. And it's a fantastic opportunity. And so if I was someone thinking about research today, I would think randomized trials is the most exciting area. It's the end product of all of this really clever science of how do you treat patients better. And it would be true not just for drug therapies or for interventional therapies, but also for screening strategies, for how you roll out vaccines, whether you use boosters, when you use boosters, etc., etc. So I think fundamentally it's a fantastic area to think about. It's data science, it's delivery, it's understanding the biology, understanding the epidemiology, so you're asking the right question in the right way. And the great thing about it is you run into the dentist with a result that will get implemented tomorrow and save lives.
Great. I mean, I think that's a perfect place to end. You've been incredibly generous with your time. Thank you so much for all of those thought-provoking stories, Sir Rory. Thanks.
Pleasure. Thank you very much.
Sir Rory, thank you so much. You've been incredibly generous with your time.